So I'll be talking to you about plant immunity and plant pathogen interactions, and I'll try to get to the topic of plant pathogen interfaces. Before I get to that, I want to, just to get everyone on the same page, I want to give you a bit of an ABC of plant pathogen interactions, plant immunity. So first of all, plants have an immune system. Not everybody appreciates that, but they do have a very effective immune system, as you can see here on this slide, if I get this right. Oops, sorry. If you can see here, this plant happens to have a single gene that codes for an LR-type immune receptor, and that makes it resistant to the potato blight pathogen, Irish potato famine pathogen, compared to the two neighboring genes. So these immune receptors detect proteins that are called effectors, but what we now also appreciate in plant pathology is that pretty much all pathogens, viruses, fungi, bacteria, nematodes, aphids, and so on, secrete effectors that act on host targets and modify host processes in many ways that help the pathogen cause disease. And I'll give you a couple of examples later on of how they operate. But plants have these immune receptors, and a single immune receptor can basically detect one of these effectors, which can be really delivered by the hundreds, by the way, depending on the pathogen you have. And in those cases, you get disease resistance. So that immune receptor will basically essentially overcome the action of all these other effectors and result in resistance. A very dynamic system, and you might be wondering, what's the problem? Why don't we just make our crops resistant? And the real problem here is that the immunity is very much nullified by evolution, by pathogen evolution. So we continuously have new races of these pathogens that overcome virulence in really same way we have these COVID variants and the SARS-CoV-2 variants that overcame the vaccines. Our plant pathogens are continuously spewing out new races that overcome immune receptors by essentially making variants of all these effectors. An incredibly dynamic system, and if you look at the genomes of these organisms, which we like to do, you will find that the effector genes and the immune receptor genes are actually the most diverse gene in both types of organisms, as you would expect with an immune system that's engaging in arms race coevolution. Okay, so that's my brief introduction. The opportunity we have, the research opportunities, is actually massive, because we now reach the stage where we have really a deluge of plant genomes, and high-quality genomes too, because really this is sort of plant genomics 2.0, because in the early phases, a lot of these very complex gene families were really just not properly resolved. But now with the newest technology, we can actually take, for example, the Solanaceae family. So these are actually the potato, tomato, pepper, eggplant, and so on species. So that's just one family of plants. So we have over 300 high-quality genomes. We can reannotate all these genomes, 15 million proteins, and about 1.3% of those happen to be immune receptors. So that tells you the diversity and the extent to which plants actually devote to their immune systems. Over 1% of all their proteins are basically involved in immunity. And actually, to date, we don't really have all the functions for these NLR proteins in plants besides immunity. So we end up having this huge diversity of about 200,000 NLRs just with this one family. We can use phylogenomics to classify them, and these days also we can use AlphaFold and all of that. But really the challenge, and in a way the focus of my lab, if you want to sort of bring it down to the basics, is making sense of this gene alphabet soup, gene or protein alphabet soup, where we sequence plant genomes, we sequence pathogen genomes, and we have a lot of these mixtures. And then can we use different tools and different methods to really predict as much as we can out of the sequence. And I'm not going to talk about that today, unfortunately, because this is work in progress, but we did receive funding from Google.org, the philanthropic branch of Google, to really make predictions from pathogen and plant genomes and identify as many interfaces as possible. That work is ongoing. It's not ready for prime time, if you like. But what I will do is I will give you examples that will illustrate really the ultimate goal. And I think the dream we have really is that from these sequences, we'll be able to predict every single interface that we have between a pathogen protein and a plant protein, ideally receptors, and there must be really thousands of them, and also just do that only from the sequence. And I think it's something, if you told me about this a few years ago, I would have said, get out of here, it's impossible. But I think I start to kind of get a sense that we're now on the cusp of being able to do that. But that would be really for a talk for another time. What I want to do now is go back to these NLR immune proteins and give you a little bit of basics, and then we'll go back to pathogens and microbes after that. So NLRs, what are they? So these are multi-domain proteins, receptors, and now we know that they're everywhere. We carry them. People with Crohn's disease, for example, have mutations in an NLR gene that makes us hypersensitive to normally harmless microbes. And bacteria have them, fungi have them, et cetera, et cetera. So they're across all kind of kingdoms of life. And they essentially function as switches. We heard about switches earlier from Keir, so they're really staying as in an off state, in an inhibited state, sometimes auto-inhibited. The one we study forms a homodimer that's inhibiting itself. And that's absolutely off. But when they activate, there's an ADP to ATP switch here also. And they oligomerize into these structures that we in plant biology call resistosomes. A very poor terminology, I have to say. We should call them inflammasomes because functionally, really, or structurally, they really look like inflammasomes, the ones that are involved in human immunity. And the outcome of this oligomeric state is cell death in plants. So a hallmark of the immune response in plant is a cell death response. Now you can understand why they have to be auto-inhibited or maintained in an off state, because you cannot afford to have a little bit of leakage in terms of activation. I'm not gonna, I'll show us a couple of slides, but just for your own information, when you see this brown kind of response on the leaf, that's basically our readout for an immune response. And that also results in blocking pathogens from spreading and infecting plants. That's the resistance, that's a virulent race, and that plant carries one of these immune receptors. Okay, so I just added this slide at the break. I realized I should really put it because one of the beautiful things of biology and one of the beautiful things also of structural biology, we heard that earlier also from Kirill, and starting to see really similarities beyond what we could see at the sequence level. And this is illustrating that now we know that these inflammasome type oligomeric NLRs occur across all kingdoms of life. So we have here E. coli, so in bacteria, they're also involved in immunity, but they can be involved in other functions, as Susan will probably tell you. And also in mouse, for example, we have some pretty famous inflammasomes like the NAP2 and LRC4. This is one of the plant ones. In plants, at the moment, they're thought to form channels that go into membranes, primarily the plasma membrane, but we have a paper with my colleague Tolga Bozkurt coming out soon on how they also go to the chloroplast membrane, and some of them subset of these. And at the moment, there is a calcium channel activity has been described for this, but I think we can all expect many more activities and functions for these membrane pores that basically define plant immunity. So why not? I'll show you an example of one of these. This is actually a structure we solved over a year ago in collaboration with Michael Webster. This is a hexameric resistosome, a plant protein again. And this is, in fact, the AlphaFold model, which turned out to be, to our maybe disappointment or not, I don't know, you can view it in different ways, incredibly similar to the experimentally solved cryo-EM structure. You can use this little trick of adding oleic acids, and it does place it exactly where the membrane should be. Okay, biology. One of the beautiful things about biology is always diversity. And indeed, we have a lot of diversity. When the first structure of these resistosomes was solved, late 1990s, late 1990s, late 2000s, 2009 or something like that, then everybody thought they will be very similar, but they're incredibly diverse. And that's what we start seeing here already in the phylogeny. There's a lot of different clades. but we also have a lot of different structures, both in the resting states, which surprisingly turned out to be more difficult to get at, and also in the activated state, where we have different also stoichiometries. We even have an octamer recently solved in collaboration with Jonathan John's lab. And these tend to sort of be reflected by the phylogeny, where we have a lot of diversification in different type of resistosomes. Okay, so what I want to now talk about is how we actually got into the business of studying these NLRs. We actually started from the pathogen side. So this is Shihang Wu, who's now at Academia Sinica, running his lab there. He was studying this effector from the potato blight pathogen called AVR-BLB2, and we knew it was matched by a potato gene called RPI-BLB2. And what Shihan was endeavoring at is discovering what was downstream of RPI-BLB2. And he was fortunate because, as you can see, there's no redundancy here. There's only a single gene that's downstream of RPI-BLB2, and that's an RC4. And it turned out that actually these are all NLRs that are phylogenetically related to these sensors, but genetically they function downstream of these, what we call sensor NLRs, or what plant breeders define as disease resistance genes. And as you can see, they form a network which is quite complex in configuration. You have many to one or one to many, so it's truly a network, a bow-tie network. I'm not going to talk much about that. We have been working quite a lot in understanding how this network has evolved, how it functions, how sensors activate the helpers. Let me simply say that these are the guys that form the resistosomes and execute the immune response. These have actually subfunctionalized and lost the capacity to oligomerize. Their job is simply to detect the pathogen and activate the downstream helpers. So from a common origin, a common phylogenetic origin, we have specialization and subfunctionalization of these NLR proteins. However, when we discovered the network, we immediately thought, okay, this is a bow-tie network, and these are obviously the nodes. We have a lot of respect for pathogens and microbes, so we said pathogen must have evolved ways of targeting these nodes and suppressing them. And so Lida Derevnina, who's now continuing actually this type of work in her lab in Cambridge, she set up a screen and collected a lot of effector proteins from many different potato pathogens and screened them for suppression of this network. And I will talk about two of them that are actually where we have now the precise function, but Lida studied actually additional ones. And this is maybe my moment to make a little comment here and maybe ask you for a quiz. Do you think that parasitic nematodes should be considered microbes or not, and relevant to this conference? Because to me, they're actually tiny enough and microscopic enough to be maybe viewed as microbes. So let's, for the sake of this talk, let's treat a parasitic nematode as a microbe, okay? So please bear with me and let's not get into the semantics. They're very, very tiny. So the first example I will talk about comes from a parasitic nematode. This is the potato cyst nematode, which secretes a tiny protein. This one's really small. We call SS15. And it turned out that SS15 directly binds these NRC nodes and inhibits them. Let me show you how that looks like. This is one of these helper protein. It sits as a homodimer in the cell, auto-inhibited. It activates into this hexameric resistosome. But when the nematode comes, the nematode protein binds the homodimer and prevents it from opening up and oligomerizing into that hexamer. In fact, it binds right at the hinge where there is a rotation of the NB domain and prevents it essentially from opening up. So that's a very elegant example of how these parasites evolved to inhibit and suppress these immune receptor proteins. The second example is from maybe a true bona fide microbe. It's the potato blight pathogen. This is an oomycete, fungus-like organism, if you like, eukaryotic microbe. And in this case, we identified a protein called AVR-CAP1B, and I'm going to show you how this protein also inhibits the resistosome, and I'll show you this through this animation. So that's what happened when we have activation of these NLRs, is NRC3 in this case, one of these, again, helper nodes. It oligomerizes in a stepwise fashion. And what the effector does, it actually grabs it while it's oligomerizing, it inhibits and blocks the assembly process. So it binds to the trimer, if you like, or to an assembly intermediate. And the effector is quite a large protein that's L-shaped and actually wraps and makes contacts with 3 of the different protomers. And that's essentially exactly half of the resistosome, and that contributes to inhibiting this protein from functioning. Okay, so those are 2 examples, I guess, of pathogen-plant or effector-receptor interfaces. It's really satisfying to me that this is very sort of basic fundamental research that has actually led to very obvious applications. Because if you think about it, if these pathogens carry inhibitors of these nodes, they're actually shutting down a lot of R genes that are present in our crops, right? And so these R genes many of them will not function because we have these inhibitors and suppressors. So we reasoned that if we take one of these helpers and mutate that interface, because we know the interfaces, that's the value of actually knowing the interfaces, we can introduce point mutants and really resurrect these genes that are normally inhibited and suppressed. So we did this already as a proof of concept with the nematode effector. So this is X15. Bear with me, this is a bit of a complicated experiment. This is actually the resistance gene and the other nematode protein that are detected by that resistance gene. So normally you get an immune response when you have the helper being provided. That's the control, if you like. But that response is suppressed by the nematode inhibitor. If we introduce a point mutant in that interface that the inhibitor binds to, just 1 amino acid change here in this position, then the resistance gene is not inhibited anymore. We can essentially, we call it resurrect this gene to function again. So the idea here is that our crop actually are carrying a lot of resistance gene that need to be upgraded to be basically brought back to life, if you like, or resurrected. And that's actually very exciting in the context of the Precision Breeding Act, the gene editing technology, etc. So that led us with my colleagues, Kian in particular, who's the CEO and co-founder, and also Tolga Bozkurt at Imperial College, to start this company, Resurrect Bio. And Resurrect, you may have seen in the news, recently closed the Series A, so we're quite excited about the proposition here of going to plant breeding company who have a lot of germplasm and telling them that they already have useful genes in their germplasm. They just need to bring them back to life and resurrect. So with that, I'll stop there, and I want to thank my lab, of course, and many people really contributed to this work. That's some of the snapshots over the last years. Many of them are here. I also want to thank Meyer alumni. I mentioned Lida's work in particular, but others also in the team have contributed more recently. I mentioned Michael also and the work with the facility here for the cryo-EM and electron microscopy, and my colleagues at the Sensby Lab. So thank you so much.